Hi everyone, I'm Dongsheng Guo. I'm now an instructor in Emerson Lab at UMass Chan Medical School. So today I'm glad to talk about my project. It's different with muscles. It's using FSHD iPS cells to modeling the patient hearing loss. So we all know that FSD is caused by DUS4 misexpression in muscle cells. So last year there is a report showed that the congenital arrhythmia is also caused by DUS4 misexpression. So this means that DUS4 still some unknown function in some disease. So for FSD research and therapeutic development, the cell model is very important. It's also the start point. So for here, as Kellen talked about, we generated a primary myoblast isolated from patient biopsies. So sometimes we can't get the patient myoblast, we only get the patient fibroblast. So from that point, we made iPS cells from patient fibroblast or myoblast. The iPS cells could maintain the genetic and epigenetic background of donor cells, and the iPS could self-renew in dish and could differentiate into 3 germline cells, including muscle cells. So here in Emerson Lab, we have generated a series of FSD iPS lines, including 5 early onset FSD IP lines. These 5 lines have shorter default repeats from 2 to 5, and 2 adult iPS lines with longer default repeats, 7 and 8. We also generated 3 non-manifesting iPS lines. We have this line have less than 10 default repeats, but no phenotype. And 3 health control and 2 FSD2 iPS lines. Recently, we developed a technology to make functional and maintainable muscle stem cells from iPS cells. This is made iPS from skin biopsy and fibroblast and myoblast. And we did muscle differentiation for undergo S1, S2, S3 stage. At the final stage, there's still some undifferentiated cells or reserve cells. And we could isolate and maintain these cells. And we named this cell is iMyoblasts, short name IMBs. These cells is PEG3 positive cells and could differentiate into imyotubes. And after the xenograft into mouse, could differentiation and regeneration. So using iMyoblast technology, we made iMyoblast from FSHD, and after we get FSD iMyoblast, we infect the cells using DUS4 GLP reporter lentivirus. So after differentiation, we could see the DUS4 nuclear here, and in the control cells, we didn't see any GLP signal. This means that FSD iPS cells is useful to model this muscle model muscle disease. Besides the muscle weakness, there's still some comorbidities. Amongst them, hearing loss is very common in patients. And also in the mouse model, the mouse still have very high frequency of hearing loss. So for the hearing loss, we still do not know how or what's induced hearing loss in patients. So we found that in general, hearing loss usually caused induced by the ear cell damage. So we thought that maybe FSHD hearing loss is induced by hair cell damage and induced by DUS4 misexpression. But in the humans, there are only 75,000 sensory hair cells. So it's very hard or impossible to isolate these cells. So we thought that maybe FSHD iPS-derived hearing loss is a good choice to investigate the mechanism. So we looked back our iPS lines. We found that we have two iPS lines from patients with confirmed hearing loss. This is 75, 74, and 85. Also is with short default repeat. So we did the differentiation in dish in 2D dish the iPS 2-ear hair cells, and we test the DUS4 biomarker. We found that the two patient hair cells have very high biomarker gene expression. Here the lower number means hair expression. So it's very promising that maybe DUS4 misexpression is the cause of hearing loss. But as we know, the hair is very complex. There are many kind of cell types. So we thought that maybe 2D differentiation can't recapture the space structure. So we thought that maybe FSHD iPS-derived 3D differentiation or organoids maybe could model the structure. So this is the cell images from a reference paper. Here is hair cells. So now we are following this protocol to make FSHD ear organoids. Here is showing the 17U is a control, and the patients is from start point to see the 3D organoids. And once we get the organoids, we want to do single cell-seq to confirm if DUS4 is induced. And then we will test the Callan's drug siRNA to see if DU01 could knock down or decrease DUS4 expression in hearing loss. So for the summary, so we made iPS cells. We made FSHD iPS cells. These cells could be used to model skeletal muscle weakness and hearing loss. This is present now. Also, besides the muscle modeling, we want to plan to make universal iPS cells using knockout several genes to make hypoimmunogenic iPS cells, and then make universal muscle stem cells to development muscle stem cell therapies. So we have a great team. So we have Judith is from my year and I and UMass Memorial could help us for the hearing project. So thank you for donating your cells. So we need to work together. Okay.
Dongsheng is in discussions with Jess Statlin, who has this pediatric study going on. It was mentioned earlier, this MOVE study, to incorporate hearing loss into the studies of these earlier onset. So we're in discussion with them to see if we can partner into that MOVE study. It's a really understudied comorbidity that's really would be important to understand the role of DUX4 and is a possibility of different kind of therapeutics other than muscle therapeutics for hearing loss. So.
Thank you.
our next presenter is Dr. Christina Shea from Spalding Rehabilitation and Mass General Hospital, I guess.